Arratsaldeon, denoi, eskerrik asko etortzeagatik. In particular, I would like to thank Professor Brenner for accepting our invitation to participate in Zientzia Foroa. Dakizuenez, hitzaldi desberdin batzuk antolatu ditugu aurten, ia bukatzeko zorian gaude etzi, izango dugu benetako aurtengo azken hitzaldia Bilbon, Guggenheim Museoan. Hasi ginen nazioarteko kimikari buruzko urtea zela aprobetxatuz Luis Oro irakaslearekin. Gero Sonia Schultz irakasleak etorri zen Bilbora, bigarren hitzaldia ematera. Gero Bilbon ere Maria Blaskok eman zuen hitzaldi bat biologi molekulari buruz, telomeroi buruz. La siguiente conferencia ya la dio aquí María Vallet sobre materiales, biomateriales para utilización como implantes en biomedicina. Esa fue la primera conferencia de Zientzia Foroa de este año en Donostia, en San Sebastián. Y hoy tenemos la segunda conferencia, que va a ser impartida por el profesor Sidney Brenner. La última tendrá lugar, como he mencionado antes, en Bilbao, en el Museo Guggenheim. La impartirá el profesor Ulrich Beck sobre qué Europa queremos. Quizás este repaso que he hecho de las conferencias de este año ejemplifica, pienso, bastante bien el espíritu del ciclo Zientzia Foroa, organizado conjuntamente por Ikerbasque, la Fundación Vasca para el Avance de la Ciencia, y Jakiunde, la Academia Vasca de las Ciencias y las Artes. Conjuntamente organizamos cada año el ciclo y ya estamos preparando para el año que viene los conferenciantes que vamos a traer. En Facebook en Facebook y en la página web de Ikerbasque tenemos habilitada la posibilidad de que los asistentes propongan nombres de posibles conferenciantes en los que podrían estar interesadas las personas que piensen asistir, y nosotros conjuntamente, lógicamente, con el comité de Jakiunde, pues analizaremos las peticiones y vemos en qué medida estas podemos, por muchas razones, desde ajustes de agendas a otros factores logísticos, pues organizar unas conferencias que sean del interés para todos. El objeto final es transmitir y compartir con el público los resultados de la ciencia y de sus valores, trayendo a conferenciantes, como podrán ver en la conferencia de hoy, de primera talla mundial. Y sin más, en este tipo de actos siempre digo que el importante es el conferenciante. Sin más, ya voy a terminar mi introducción general, y el profesor Ginés Morata, eminente biólogo, Premio Príncipe de Asturias de Investigación Científica y miembro del Comité Científico Asesor de Ikerbasque, va a hacer la presentación más específica del profesor Brenner. Profesor Morata.
Buenas tardes a todos. Para mí, presentar a Sidney Brenner, aparte de ser un gran honor, un enorme honor, es también un gran placer, y de luego hacerlo aquí en San Sebastián y con el auspicio de Ikerbasque, que realmente han hecho todo lo posible porque estuviera Brenner aquí hoy, y el DIPC también, que nos ha realmente facilitado toda la estancia y la llegada de Brenner. Pero realmente es un placer tenerle aquí. Y solamente quiero comentarles lo que representa para la biología Sidney Brenner. Sidney Brenner es seguramente el científico, el biólogo más importante del siglo XX, el más influyente, y ciertamente y su importancia ha aumentado en el siglo XXI. No es difícil imaginar para ningún biólogo hoy día alguien que haya tenido más influencia y más impacto en la biología del final del siglo pasado y del comienzo de este siglo. No voy a entrar en los detalles de sus muchos descubrimientos, ni tampoco en los innumerables premios que se le ha concedido, que culminaron con el premio Nobel en el año 2002, pero que muchos biólogos pensamos que tenía que haberse concedido muchísimo antes. O por lo menos tenía que haber obtenido otro premio Nobel anterior al que tuvo. Sidney, junto con personajes como Francis Crick, otro premio Nobel, fue el fundador de la biología molecular, que es la rama de la biología que está destinada realmente a cambiar hasta el futuro biológico de la humanidad. Yo no voy a entrar en los detalles de sus descubrimientos, pero sí quizás les interesa saber que el impacto que ha tenido ha sido enorme. Fue en los muchos años que estuvo en el laboratorio de biología molecular de Cambridge, de los años 60 hasta los años 80, y allí formaron innumerables científicos en esta nueva doctrina que ellos habían creado, que es la biología molecular, y todo este conocimiento y toda esta forma de trabajar, toda esta filosofía científica nueva se extendió por todo el mundo, y aún estamos viendo todavía, y estamos viendo hoy día sus frutos. Pero Sidney es una mente extraordinariamente inquieta, y en los años 60, a mitad de los años 60, decidió que la biología molecular había entrado en su fase académica y que él tenía que hacer otras cosas arriesgadas, difíciles y que nadie había intentado. Y decidió trabajar con un gusano, un nemátodo, que se llama Xenogratitis elegans, para estudiar cómo esencialmente el sistema nervioso, pero en general cómo se forma el cuerpo y el sistema nervioso de un organismo multicelular, de un animal multicelular, uno de los problemas más intratables y difíciles que había tenido la biología. La comunidad científica era muy escéptica cuando empezó, porque no había ninguna experiencia en este tipo de experimentos y con este tipo de organismo. Hoy día, para decir el éxito que ha tenido este organismo, hay aproximadamente 400 laboratorios en todo el mundo trabajando en ello y unos 4.000 científicos que se dedican precisamente a trabajar con este organismo, y ha dado lugar a descubrimientos importantísimos, y varios premios Nobel, aparte del de Sidney, han venido del trabajo hecho con este gusano. Esto para hacerles ver la importancia y el impacto tan tremendo que ha tenido su trabajo. Pero en los años 80 también decidió que ya el gusano estaba entrando en una fase académica. Necesitaba más excitación, cosas más novedosas, y decidió trabajar en un pez que se llama fugu, que es un pez que los japoneses comen y se arriesgan su vida cuando lo comen. Este pez que necesita a alguien que lo limpie especialmente, porque si no tiene unas bacterias simbióticas que producen una toxina que es letal. Bueno, pues con este pez lo escogió Brenner porque, por así decirlo, puede ser el genoma modelo para estudiar los genes humanos. Típicamente él utilizó una estrategia que nadie se le había ocurrido. Este pez ha producido unos resultados interesantísimos para entender nuestros propios genes y nuestra propia evolución, que es los últimos temas de los que está trabajando Brenner. Yo ya quiero acabar aquí, solamente decirles que es un enorme privilegio para tener a Sidney Brenner. Difícilmente habrá otro biólogo que pueda venir que tenga el impacto y la categoría que tiene Sidney, por lo tanto ya le dejo paso a él, que es mucho más interesante lo que él tenga que decir que lo que pueda decir yo. Muchas gracias por su atención.
Well, first I'd like to thank all the people who have organized my visit here for inviting me. to come to this part of Spain, which I never visited before. And I have to say that I've had, so far, a very good time. But unfortunately, it's been too much eating, too much drinking, too much talking. So now I have to talk again, because as you know, you do not get food till you sing for it. So I'm going to sing for my supper. I've been asked to give a lecture on what's called the human genome for an audience that is not professional, does not know any genetics, so I have to start at the very beginning. So, of course, all the professional people should forgive me for making things perhaps a little too simple. So what is the most important biological observation that we know of? Must have been the first thing that an intelligent man noticed when he walked on the surface of this planet, is that animals and plants reproduce, but they always reproduce their own kind. As we say, like begets like. So flies give rise to flies, humans to humans, sunflowers to sunflowers, and that must have struck everybody because, of course, they were observers of nature. So how is it, you might ask, that a complicated organism like an elephant can produce another elephant? Because clearly what passes between the generations is not the elephant itself, although for a long time they thought there was a little homunculus in the sperm, and all it did was penetrate the egg and then grow big. We now know it's not the object itself, but a description of the object. A description, I don't call it a program, I call it a description of the object that is handed down from generation to generation. Right? Now, what is this description? What is, if you like, the information that we hand down? The remarkable thing is that in 1953, for the first time, we could actually study information in the living world, because information is coded in chemistry. So an information molecule is the DNA sequence that lies at the heart of what you pass down. And so we had another quantity in biology. Everybody was concerned with matter. How did biological organisms make all the things inside them? Or with energy, where did they get the energy to do this? And the revolution that we had in 1953 is that we have to deal with a new concept, and a concept that is embodied directly in the sequence of DNA. And once you have that, then I think we can now start to do real science on this problem, which I think could not be done until we had the structure of DNA. Because anybody who saw it, and I saw it as a young man very early, before it was published, 1953, and I just said it was a complete revelation, because now I knew what had to be done in order that we could understand how living creatures reproduced themselves, how they copied the information. But I'm going to tell you a little story which is not widely known. There's a very famous physicist and mathematician called von Neumann. And von Neumann got very interested in just the abstract question of a self-reproducing machine. He said, how would I make a machine that would copy itself and reproduce itself? And he then proved, it's a logical proof, that you had to do it in the following way. First, you had to have a description of the machine. That description, which we can call a tape, which in fact we would call a gene or DNA, that description is then used by the machine to build another copy of the machine. And then at the end, you have to copy the tape and insert the tape in the next machine. And if you look at that, that is exactly in the logical form of a self-reproducing machine, exactly what the DNA accomplishes biochemically in the exact state. So it is an implementation of that. Now, of course, although I have to tell you that von Neumann had these thoughts, and he wrote about it in his theory of self-reproducing machines long before people knew about DNA. But such was the culture of science that nobody knew about von Neumann, including people who it is very clear actually were an audience at a lecture he gave in California in 1951, long before this. So that is how history looks as though von Neumann is the genius behind everything. But if you ask Watson and you ask Crick, they wouldn't have known who von Neumann was. Of course, von Neumann is very famous today for not only inventing, but he invented the modern computer. He didn't actually build it, but he invented the form of it. And what he said, you could represent this in what's called a von Neumann machine, because you can have a tape which you read, and of course that dates back to an earlier discovery by a man called Turing in the 1930s. That is, what can be accomplished in any machine that can be expressed in terms of a universal Turing machine, because it says it provides you with the mechanical rules for computation of the answer. Well, we must now try to find out about this tape within us, and let's just look at what it is. So it consists not of these blocks, but I mean of these letters, which represent the individual chemical entities. It's a language called of nucleotides. There are 4 kinds, called guanine, cytosine, adenine, and thymine. And there is a principle of self-complementarity here, so that it doesn't consist of one sequence, it's 2 sequences which are paired according to the rule wherever there's an A, there's a T in the opposite strand, and of course wherever there's a T, there's an A, and wherever there's a G, there's a C. So given any sequence of DNA, you can write down the opposing sequence. But the beauty is, if you separate the 2 sequences and assemble the complements on either side, you land up with 2 copies of the double-stranded DNA. And that's actually how it occurs in nature, that we copy the strand into one. And then, of course, that says how we can copy it equally. So we can copy the tape. The tape can now go and make a new machine under these instructions that are written on the tape. So in a sense, the whole of the living world provides a fascinating and extremely deep question about a number of things. One, how did this complexity evolve? How does it that we have such wide variation? And then how is the tape read? Because the tape is read in order to make real material objects. So we know that a lot of it is translated into another language. It's a language, another chemical language, except this language has 20 different things called amino acids. And each of the tape is simply translated into this. And so one of the fundamental questions that we asked right at the beginning was, how do you go from a 4-letter language to a 20-letter language? And many things were done, but of course we now know that there's a code that 3 letters in the nucleic acid language stand for 1 letter, that there are multiple representations for some of the amino acids. And the remarkable thing is that to a very large extent, the code is the same in all branches of life, whether they are viruses or bacteria or fish or people, we all have the same genetic code. So there is a unity of chain. Now, of course, when you have a code like this and you are copying it, you can make errors because, as everybody knows, nothing is perfect. And so when you make errors, you can put in the wrong base. It can be done with certain degrees of mistakes, and of course we call those mistakes mutations. And if the mutation is in certain bases, we can change one amino acid into another. So we can change the product. So we can make alterations. And just to give you some idea of what it is, there are today, I'm told, 7 billion people, 7 times 10 to the 9 people in the world. Okay? There are 6.4, I think, billion bases in the 2 copies of your chromosomes, because you get 1 copy from your father, 1 from your mother. So we have that. That means almost certainly somewhere in the world every mutation has already occurred. Of course, we can't see the effect because they have 2 copies. Right? So this means that if we could find everything, we could explore the whole potential changes that we could do by doing this. And of course, since mutations could occur and were noticed in nature, and of course we can induce them at a higher frequency with various forms of radiation or chemicals, we could actually change organisms. We could make mutants. And that began the subject of genetics, which Mendel did in the middle of the 19th century. He started to cross peas, and he had a very simple theory that how these genes, they were called Mendelian factors, and they gave rise to characteristics of the organism. Today, we would say the Mendelian factors are called genes, and the collection of them is called the genotype, and all of the DNA is called a genome. Those are just words. And of course, what they present themselves is called the phenotype. That is what the genes do. Right? And if genes can make proteins to do the work that is in the cell, and so all catalysis throughout the universe of life is carried out by these proteins. And they are remarkable, flexible things. They can do many things. And we now know of hundreds of thousands of different proteins. We seem to find many collections. Every time we explore another feature of the living world, we find curious organisms that can live, for example, in boiling water. There are bacteria that can live on hydrogen. And the most fascinating one is if you go along any highway, you will find bacteria in the soil next to the motor roads that can actually deal with carbon monoxide. They can actually use carbon monoxide and live with it. That's, of course, what's come out of the exhaust of cars and has enriched the growth of these bacteria that, of course, are hungry for many things, for many carbon compounds. So we have all of this in this vast armamentarium. For years then, geneticists tried to explore the relationship of the genetic material, which they only knew were on chromosomes. They could try and do this by genetic mapping. So those were all the great accomplishments of the classical experience of genetics. And they tried to understand how it is that you make a hand, right? And of course, we don't have an instructions to make a hand in the same way, because if you were to ask someone, how do you make a hand? He'd probably write a computer program saying for n equals 1 to 5, repeat, make a finger. So you make 5 fingers, and then you make a hand. We don't make hands that way. So the discovery of how all of this construction has now occupies the people in the life sciences. Right? So, what could we find out? Could we short-circuit everything by actually finding out what is written in the DNA? If I had the sequence of bases, which of course we tried to get by genetics in the old days, could one determine the chemical composition of the DNA? This is just a problem in chemical analysis. We call it a sequence because these are very long, very thin, one-dimensional organisms. All the DNA in your chromosome is one molecule. It just happens to be a very big one, a very long one. You have about 1 meter of DNA. The spacing between the bases is 3.3 angstrom units. So you have 3 bases per nanometer. So it really is nanotechnology. In fact, it's sub-nanotechnology, and that is quite an interesting way of looking at it. Right, so, well, Fred Sanger, he invented a way of sequencing proteins. Up to the time he did it, most chemists did not believe that proteins had a formula. They thought they were just sort of messes, globs and polymers of some undrawn. And I was at a meeting in the 1950s where the doyen of British chemists, Robert Robinson, after Fred Sanger gave his talk on finding the sequence of insulin, which was the first one he did, and said, this is remarkable. It means that proteins have a chemical formula. Like I can write down the formula for urea or for any one of the chemicals that we were doing. You can write down the formula for a protein. You could also write down the formula for a nucleic acid. So happens to be called a sequence, but please, it is a formula. And you can synthesize it if you want to. People have synthesized protein. Today, people have synthesized DNA. That's called now, a new name is called synthetic biology, but it's not, because all they're making are forgeries. They copy another piece of DNA and they put it inside a bacterium, which means that they have forged it. They haven't invented anything. They've made a copy, and of course they're not very good counterfeiters because they put their names on this as well, so we can catch them very easily and say, that's not a real gene. I mean, that's just a copy of a gene, and so it's a counterfeit. It's not the real thing. However, well, so now there is this following challenge that we can actually decompose the huge molecules, the huge complex genomes, and we can find the sequence of them. And when the first organism, you have to realize, that was completely sequenced of any complexity was a small virus for bacteria called lambda. It had 45,000 bases, 45 kilobases, as we call it. And at that time, people said, what if we could sequence the human genome? Well, of course, that was a tremendously huge jump in magnitude from 5 times 10 to the 4 bases to 3 times 10 to the 9. Yeah, 5 orders of magnitude, just like that. And of course, it was a challenge, and people then decided that this is what they wanted to do. But first they tried smaller things like flies and so on, and gradually we worked up the scale till the genome became possible to sequence. Now, of course, there are 2 ways of doing this sequence. One is what I call 3M. 3M is a famous company in America. For us, it stands for machines, money, and management. That is, you can set up a big lab, and I, in fact, once suggested that we should set up a penal lab so that everybody caught cheating, copying other people's work, would be sentenced to this lab. It would be in a very undesirable place like Chicago, and he would be sentenced to do 1 megabase, that is 1 million bases of sequencing. But that was just a joke, of course, but was taken quite seriously by some people. So eventually, by huge, it was calculated that it would cost about the same as the number of bases in dollars. So it would cost about $3 billion, which today sounds trivial, peanuts compared to the trillions that people are talking about. As I read a book by one of the people who was the treasurer, he was a treasury secretary in the last Labour government in Britain, and he was worried because he had to produce something like £5 million to save a bank. He said little did he know that in a few months he would be writing checks for £50 billion. And these numbers now don't mean anything, few trillion here and there. I mean, what does it matter? So, but these were real numbers. You had to do it. So, of course, it required either some new advance in technology, so it could be done very simply, or it required some unorganized activity. It also required raising a lot of money. And so it became very similar in the way people saw it as to put a man on the moon. Also cost about $3 billion. And they decided we would like to do this by the end of the century. That is, by the year 2000, we should do this. Now, in a curious way, it was very similar to putting a man on the moon. Because if you think about it, it's very easy to send a man to the moon. That's trivial. The problem is, how do you get him back again? Now, that's the expensive part, is to get him back. So, in a sense, it was very trivial to sequence the human genome, which is now, so to speak, stranded on a moon there, and now we have to find out how to get it back. We have to interpret it. We all knew that that was going to be the biggest task of all, because we had no experience in encountering such a mass of information. Well, as many of you know, when it was sequenced, as I used to say, when Tony Blair and Clinton announced they had sequenced the human DNA between the two of them, and that it was all done, they left the next job to us scientists. But everybody said, this will be the great message that they can bring to everybody. We will have new drugs, we will solve all diseases, we will solve all human problems. And that was, I think, as they say in the trade, it was a bit of hype. That is, it was exaggerated, because there was nothing like that in the end. And the 10 years was celebrated this year, actually the end of last year. And people are saying, well, what actually did you deliver for this? And we have to say, not much. But one of the things is we just had no idea, and there still is not a good idea, not good ideas on how to deal with the interpretation of this information. So the first concept I'd like to tell you is the concept of junk, right? And I must just define that very carefully. See? Because if we said every base in your genome is important, which it is in a bacterium, because every base counts, if it was important, how many genes would you have? So we began to think, well, how many genes does it take to make a complex thing like a human? And what we knew is we had sequenced bacteria. In fact, the common bacteria of your intestine, E. coli, it's got 4,000 genes. So everybody thought, well, I'm much more complicated than E. coli. I must have many more genes than E. coli, right? And so the guesses must have hundreds, thousands. But if your genes were the same as E. coli, we can make a calculation about how many genes you might have had. There's something called, it's a short gene, it makes an average protein. Give or take, we would say, you must have, if they were E. coli-like genes, you must have 3 million genes. That sort of sounded okay, I mean. But then, of course, we could find other genomes which weren't so big. And in particular, by looking carefully throughout nature, we found genomes which give rise to quite complicated organisms called fish that share a lot with us. They have the same genes as we do, and they had 10 times less DNA. So were you going to argue that a fish has 10 times fewer genes than you as a human? And it then became very clear. Of course, you could argue that. You could say, I'm much better looking than a fish. I must have genes more complex. So we've got to a stage, I think you can see, where we're beginning to wonder, what do we mean by complexity? And what is this measure of complexity that we've got to deal with? And do we mean that because I'm much more complicated than a fish, I just have to have lots and lots of more complex genes? It's not correct, because it is made up of actually processes that are gene assemblies, that is, where proteins work together, and therefore by doing this, they're like, if you like, subprograms in a text there. You don't have to represent everything at the common thing. And of course, we used to speak very loosely of a gene for this and a gene for that, to speak for a gene for blue eyes. And we now know that is got nothing to do with blue eyes. It's got to do with a chemical process that moves certain amino acids across membranes. So that's the real representation. Well, in order to try and explain this in more detail, let me consider the following argument. Right? You know, we have our first cousins are chimpanzees. And our first cousins, we have genomes that are very similar to ours. 99% is the same. We only differ in 1% of the DNA. So everybody immediately could argue, when we sequence the human genome and we sequence the chimpanzee genome, we will find the human has one extra gene. And that gene will be the gene for language. In other words, we're going to represent all of language in one gene. All right? You can see that this is not a correct argument, because I always used to have an alternative argument for this. For example, suppose that chimpanzees during their history found that talking gets you into trouble. They would have evolved a language suppressor gene. In other words, the language would have been a natural process, and they would have suppressed it. And of course, they would have the exact gene. So whereas, according to the one model, we would have the one gene for language, which we would call the Chomsky gene in man, according to the Brenner theory, it would be called the Chomsky gene, because they would have the extra ones. So, in fact, in order to talk about function, you have to know what you're talking about. And I think that this is still very, very badly understood by people. They think there are genes for schizophrenia, genes for cancer, genes for homosexuality. But you could also ask, maybe there are genes for luck. Maybe you can have good fortune because you've got... It's not represented like that at all. So we must really get to grips now with what the genes do within you. Well, that's the first thing. So we need another definition of a gene. It hasn't got to stand for a character, abstract character like this, but it has to stand for a process of some sort that is, in the end, chemical. That's what happens inside your body. Could be an enzyme, could be a transport program. And that is displayed in the real world in terms of a complex function that you can perform. And that is the task that we really have to have and do. Now, of course, most people are interested in the question not so much of the fundamental biology, which I just touch on for the moment, but much more is, how's it going to affect me? How's it going to affect my health? Is it true now that we know all of this, we'll be able to cure any disease and do anything like that? And the answer is, it's still a very long way from that. And the reason for that is we've got completely the wrong perspective about the human genome. If you were a biological entity, you are at the mercy, so to speak, of your environment. If you're a dog or a cat or something like that, an animal, a plant, you're at the mercy of your environment. If your environment changes and you can't actually adapt to that change yourself to adapt to that change, then you become extinguished, you become extinct, and nature just tries again with something else. So imagine we humans at a time when the world was freezing, the ice ages were coming on. in order to survive properly under those conditions, they could only do it, as many of the animals did, by growing hair, by growing a coat that they could wear. But humans didn't have to wait for that. They had another tool, which is the brain, and what they did is they went out and killed an animal and put on its skin. There you are. Huge process that would take thousands and thousands of years of biological evolution to try to grow hair on your body to keep warm. They just did it overnight by taking it from the animal. Right? And so for the first time, and I give that as one example, there are many examples like this. Because we could cook food, actually, because we discovered fire. We could actually cook food, and we could actually make things palatable that would be impossible for us to eat unless we had very special adaptations to this. So we could widen our diet. We became omnivorous. And by this second process, which we can call cultural evolution, man then came to change his environment. He didn't like it. If it was dry, he said, I'll bring water here. And of course, that is now the history of humanity as we know it. But biologically, humans are still the animals they used to be before their brains got bigger. They're still the animals, and what our body has adapted to is the following. Just to give you one example. The body says, whenever you get food, eat as much as you can. Eat everything, and then take the food, convert it into fat, and store it. Because it turns out fat is the most efficient way of storing energy that you can use later when there is famine or the animals disappear. It means you will have prolonged survival. So our body developed purely biologically, certain parts of our brain, a internal state that actually contributed to our adaptation and survival in conditions that now no longer exist, because we can change them. So if you like, we've got inside of us a genome which is probably several 100,000 years old, evolved under the conditions of the planet several 100,000 years ago, with no supermarkets, no cars, everything like that. We got all this biology, and now we're living in an environment to which it is completely maladapted. And I think that is the critical understanding that we have to have about our condition in the kind of culture that we live in now. And in fact, this is the whole history of humanity, which we need to understand fully, from how they changed from being small bands of people that were hunting or collecting things to then this organized society that we have now. And as you know, these things then change very rapidly, and of course we can now begin to see what is the most common disease of culture. It is the epidemic of obesity, and this means that the number of people who have type 2 diabetes is rising. It's rising fast in China as they begin to live in a society which has different conditions, and it's rising fast in India. Right? And it is just inevitable. It is the history of our own that's now not adapting to this. you ask yourself now, how can we adapt to this? Right? And in order to do this, I'm going to give you a semi-serious way of dealing with the disease of obesity. As of course, this is worrying everybody that there's so much loss. So I have a very simple solution to this, which has tremendous benefits for everybody. So all you do is you declare to be obese is a crime. Okay? If you are fat, you get arrested. You are then sent to my jail. In my jail, you are put on a bicycle and you exercise and you put on a diet. And the bicycle powers a pump which pumps water, because this is a good way to pump water through many, many small pipes, because there'll be lots of criminals, as you can imagine, up to the top there, and then we let the water run out and we generate electric power. All right, so we have converted biomass, okay? There's a lot of biomass walking around, especially in country. The fattest country in the world is America. The second fattest is Australia, and so on. And everybody will soon do this. And you can begin to calculate, how much energy is just wasted in this. Right? Then when you are allowed to go out, you've gained a few years of life. You're much healthier. But since we don't trust people, you have to report. You only put on parole. You have to report to the police station every month to be weighed. If you put on weight, back you go. Now, I know this is a joke, but some people think it should be taken seriously. And that says, what is it that we're doing here? We are compelling people to actually live in a world they've since deserted. They don't do any exercise. They don't walk around. They don't spend energy on this. But they still are consuming vast numbers of calories. And that needs control. Right? Now, let's just ask ourselves, how does the body actually work? In your brain, you have a region in your brain called the hypothalamus. I'm just going to tell you something about this hypothalamus that's very interesting. I take a rat and I put it in a cage and I show it through a glass wall, I show it a cat. And the rat does nothing. But I open the cage and let the rat smell the cat. And it goes crazy. It tries to run away. It's just completely fear-driven, and it needs to get away. Right? And that's actually wired into the genes here of that rat. It's wired in that it has a connection between the olfactory, the organ of smell, that goes back to the brain, and it's wired in its hypothalamus to these things, so that it results in hormones being produced and in this wild action of flight. Right? Now, of course, I can very quickly condition the rat, so now it can see the cat, and that will make it also do this. It associates the smell. This is a very classic way of teaching a rat to do something. I take out the rat from this and I destroy a tiny region of the brain called the postventricular nucleus. It's just tiny. It's maybe 3,000 neurons there. And I'll put it back in the cage, and it now smells the cat, sees the cat, walks out in front of the cat, no response at all. What's interesting is now the cat is confused. The cat has to learn a new part of its environment. But the point is this: we have these things of fear, of all this, the emotional life, is wired into us through our genes and our brains. And I will tell you another very interesting story exactly like this. In the southern states of America, there are two kinds of voles. Vole is a rodent. And one kind of vole is called it's one or the other. They're mountain and prairie voles. Now, one vole is very faithful to its mate. I think it's the mountain one. And shows the mate, and it shows what they call technically high affiliation. And the other one is just out looking for other lady voles all the time. They then found out that these voles are actually have a different kind of pattern in the parts in their hypothalamuses, right? And it deals with a hormone called vasopressin. They then found that the gene that makes vasopressin is different, has a different structure. They could take out the gene that made vasopressin and put it into a mouse. Just gave the mouse the extra gene. And immediately the mouse not only adopted a new structure in its hypothalamus for the distribution of this peptide, but actually showed a much higher affiliation. So a character like that is transferred by one gene. Okay? And that's a remarkable thing, that one can generate something that one would think is very complicated, but we have to code it very simply. And all we coded is into the control region of this gene, and what we think it says, keep on making vasopressin, make more, because that increases affiliation. So I think when we finally dissected all of this, we are going to find astonishing things about ourselves. And in a very funny way, Freud was right. You see, this bad part of ours, the frightened part, the one who's greedy, wants to eat all the time, Freud called it the id. The id is in your hypothalamus, no doubt about it. All of you have got this. But one interesting thing is you have a frontal cortex in front of this, which sends down stimuli inhibiting the hypothalamus. Right? And the technology exists to change the frontal cortex. It's called education. You can talk to people and you can change them. And so I believe that we are going to find what the genome will teach us in the new world of biology will be a new kind of public health. A new kind of public health in which we come to understand ourselves more. And that we have to say to people, you have to take responsibility for your health, your own health, right? And we must educate the people to do this. So I think if we were to look forward, as I often say, I hope they let me come back in the year 2053, which is the 100th anniversary of the DNA structure. I don't know whether I'll come down or go up. Doesn't matter. But I just want to come back for one day to see. And I think one of the dreams that we can have is that by understanding this intricate relationship between ourselves, our societies that we've generated, the way we evolved to understand our bodies in reference to this, by understanding this and being able to impart it, we can, I think, create a new kind of public health and a new kind of living for us. And by understanding that we can't just take the environment and do what we like. I think that that is what is going to be necessary in order that we can still continue to occupy this planet. So it's a long way from sequencing a genome to thinking about the fate of humanity, but that's the great thing that I think will also come in science, that we will be able to understand how all these genes that we have, how they work in order to build this very complex structure that is unique to man, which is this big brain. And I think that we have to come to terms with that. Well, I hope that this explains to you not so much the details of what we should do, but I think the details of how we should embed this new biology in what I call the new culture. And I think I once decided that what I would do is I would write a curriculum on what I call the human sciences. That is, to understand, we would call it anthropology, but that has very complicated things in academia. But the study of man and his works, and especially what has happened in the last 10,000 years. And I think if we imparted that integral knowledge together with science, the knowledge, the history of technology, is like evolution for us, because it changed the environment, and now we have a genome that's not adapted to it. Then I think there may be some hope for humanity. And as a final word, let me say the following. I was once asked by an audience, is biological evolution over for humans? So no chance of us evolving. Let us say it's a tremendous disaster, and we are reduced from 7 billion to 70,000 people, or 700,000. Would selection begin again? And I thought about this, and I said, yes, it would. We would select for small people. Okay, small people. That is, I would be very successful here because I'm very small. And if I had an American audience, I would already condemn the first row to extinction. They would all go. The people who have not seen their feet for the last 20 years, I don't know what they look like, all those people would be destined. Because, you see, you don't need so much energy if you're small. You don't need so much mass. You could have cars which were tiny if we were all very small people. But I think we would not get to that point, because I think we have to say biological evolution is over, and we must now take our future into our own hands. Thank you very much.
Thank you very much, Cindy, for such a wonderful lecture. Si hay alguien que quiera hacer alguna pregunta, pues que la haga. Yo intentaré traducirla. Are you prepared to ask some questions?
Yes, I'm prepared. I'm even more prepared for some answers.
Okay.
From you. Yeah.
So nobody seems to be.
Oh, no question.
No questions.
No questions.
Okay.
Well, it's all done.
Oh, there's a person.
Yeah.
Okay.
Okay.
You mentioned the problems of
Yes, wait for.
You mentioned the problem of coming back from the moon, right? And so there is this big deal of information coming out now from whole genome sequencing. For instance, this International Consortium for Cancer Genomes, etc.
Yeah.
So I guess the problem we have now is how we get some information of some terabytes of genome expression, etc. So, what's your impression on this? Do you think these efforts will help us?
You see, it is this great propensity of now that people can make descriptions of everything. You see, it used to be that science used to not to be data-driven. This is the new thing. They all want to do data-driven, just describe everything. We can, with sufficient money and sufficient people, we can give a complete atomic description of you. Every atom in your body we can list and write down and say, that's you. But it wouldn't be you. See, it's not even, as if I can coin a phrase, it's not even knowledge, let alone understanding, you see. Because I think the best way to think about that is the following. The animals on this planet, that's raw data. And of course, many people go around in jungles looking for different insects and recording them. That's data. Right? Linnaeus imposed an order on this, a classification. Right? And that is knowledge. But it had to wait for Darwin to come so we understood how all of these forms could be related to each other. Had to wait for Darwin and genetics to understand that. So I don't believe, you see, that this will be the case. I mean, if you can't do it, the essence of science is the following. It is to be able to predict. It is to know enough so you can compute the rest, right? Whereas the attitude now is more is better. My answer is the least is best. The least you know that allows you to compute the rest. And in fact, it's only that will give you complete knowledge about anything. So I think this data-driven science, I call it the following. I think this is omic science, as we call it. I call it low input, high throughput, no output biology. So that's the thing, you see, because you would think that you have to think. It's become a substitute for thinking. And of course, as you know, everybody now relies on computers. In fact, the dream is we'll have terabytes. Of course, I have different names for these. To me, a petabyte is a Rottweiler, and a terabyte is a bull terrier, because both are pets that bite you. So, anyway, I think that this dream that somehow we'll have enough data, and then there'll be a clever computer program that'll work it out for us, is not possible. Because computers are stupid. They are really stupid. And it is much better to combine human intelligence with artificial stupidity than to try to do it the other way around. And I think that that's the mistake now. And I'm completely convinced that, by the way, this will all go on and there'll be all of these things. But if you look at the history of science, even in the modern age where it's been organized, the big breakthroughs, the big transforming thing, been created by very small groups of people. Sometimes, often one person creates the change. Okay? And I think this big corporate structures of science, with a massive onslaught, and the organization can't create anything new because all it can do is regress to the mean. And as we know, the mean is very mediocre.
Well, I have another question. I now worked for Science World, but, well, in the recent past, I was working for the dark side, let's say. And I'm an economist. And you mentioned that the easiest part of the problem when talking about research and science is getting money to make it move. Well, from my point of view, money is the hardest part because you always have people willing to work for nearly nothing. I mean, you've got the manpower to put science in motion, but not always the resources to make it possible. And I think it's our own fault as scientists because and most of the time, we seem to be unable or incapable of convincing people of how science or certain science can be useful for them. So we fail as salesmen. I mean, we are unable to sell science to the common public.
Yeah. Well, I think, that's the endless problem that you always say is, I used to say, if you build a new institute, the first department is the Department of Bank Robbery. But that's not a good recommendation because even the banks have no money these days. But I think, you see, this is the problem. And we might ask ourselves is how are we distributing, this? And is it sustainable that each investigator should have a lab of 10 people, who are going to be students? And are they going to be, you can quickly calculate, very quickly everybody will be sequencing DNA. There'll be nothing else to do because it's not sustainable. So we must have alternative things for scientists. That's why universities are terrific, because there's so many jobs in universities that, they can teach or they can sit on committees, they can form new committees and so on. There are functional jobs. So I think the idea is not going to be feasible. There are very few people that remain productive right to the end. So maybe you should change your job. You should do science when you're young and don't need much money. You can rely on everything, and then you should become a banker or something like that. No, but I'm being serious because I don't think it's built on a structure that's like a medieval guild workshop, okay? Which is novices learning from skilled craftsmen. Now, of course, it's become very corporate in its structure. And I think that's not sustainable. It'll have to be different. But I don't know how to change this. I can only give you one thing. I was once asked to give a talk to an organization that dealt with, biotechnology, medical research, and so on. And they wanted a title, so I gave them a talk, the title. The title is called The Casino Fund. And the Casino Fund is the following. It says everybody who gives money for science gives 1% to the Casino Fund and writes it off, forgets about it. Right? That means, who do you give the Casino Fund to? You give it to gamblers who have been successful. You give it to me. Let me choose the projects. Let me choose the people. All right? Well, what was the outcry from the audience? All the business people said, how can we ensure payback on the 1%? They've got to have that, must be. So I said, okay, make it 0.1%. It doesn't matter. That they needed, and of course all the academics said, no, we must have peer review. So, as you know, peer review doesn't take any risks. It is said that every grant application can be structured as follows. One half of it has already been done, and the other half will never be done. So I think it's just the thing. And I also noticed one other thing, and that is the following. As the total money for science shrinks relatively, the total number of people allocating that money rises. And there are many committees, many panels, working groups, all doing, and of course everybody wants a measure, a metric that they need to apply. And that's all the people want. And I think that it's good because it's quite a good equation, because when there's no money left for science, then all the scientists can just allocate no money. They can divide it up amongst themselves so they can be busy. It would be, of course, imaginary money, and we have to go on to the complex plane then. But basically, I think that is a job. I don't know what the solution is. I think everything can be scaled back. You don't need a group of 16 or 40 or 38 to do this job. It's based, the Americans introduced a new form of slavery. It's called graduate students. It's true. And a new form of indentured laborer called the postdoc. Okay? And that's how they do the work. Whereas it would be much better to have a smaller group of professional research officers, people who knew how to do things, people who were skilled men of the craft, rather than employ students to do all these jobs for which they're totally unsuited for. They should be learning how to be scientists, not how to obey the master and produce this. But I'm afraid those are cultural changes that'll take a long time to change. But we need to think about this. And maybe you should ask yourself, maybe we test everybody by putting them in a room with a few pieces of wire, pencil and paper, and see if they can make science out of what's in the room. Not that they need this machine and that machine and so on. So I don't know what the answer is. And, I'm not in that game anymore. Yes, by the way, that's why the human genome, to study it, is very easy. You don't require any money. All the data's there, it's free, it's been done. Okay? You just have to use this piece of technology. It's in your head. And you don't need to be supported by anybody. The only time you need support is when you try to read a journal. Then they demand $32 from you. But that's another crime. Okay.
Well, thank you very much.
Yeah.
Bueno, antes de que se vayan, quisiera recordarles el acto que tendrá lugar en esta misma sala el próximo viernes, Fronteras de la Ciencia, que comenzará a las 4 de la tarde y al que están invitados. El programa es el que ven aquí y ha sido organizado por el DIPC, por la Universidad del País Vasco a través de Euskampus, Gizarte Kutxa y Naturgas, que patrocinan este acto dentro de las lecciones Kutxa organizadas también desde el DIPC. Repito, están ustedes invitados. Gracias.